So good morning, everybody. Today we are going to cover chapter 17. It's evolution and diversity of animals. It's a long chapter, so we'll cover it in two parts. We will be going over different animal phyla. We will be putting the animals in different groups and how we will also talk about the history a little bit, how animals came around. Usually students like this chapter a lot more than the others because they can relate to animals. Some of them you already seen around, you already know, but some of them might be new to you. So it's a lot of memorizations in this chapter. So let's talk about some common features that all animals have. So they have multicellular bodies. We are multicellular. We are in kingdom Animalia. Eukaryotic cells that lack cell walls. So plants, they have cell walls. Animals, no cell walls. If you take our cell and put it in a hypotonic solution, it's going to swell up and explode because there's no cell wall, whereas plants, they have cell walls. Heterotrophic, by ingestion, we eat other things, or animals eat other things. It's very diverse. There's a lot in this kingdom. And then we will also talk about embryonic development. So because we're trying to put them in different groups, we are trying to look at some of the differences, and that includes differences in the embryo. So blastula is a hollow ball. I'll talk about it a little bit more when we get there. So it's a stage of development in the embryo. We will be looking at that as well. And then the last one is cells bind to extracellular matrix. So our cells are bound together, animal cells are bound together, and it's bound together by desmosomes. We, I think, talked about that in chapter 3, or tight junctions, gap junctions, anchoring junctions, and that's what's making our skin watertight as well. So cells bind together. So you can just think of skin cells binding together. So those are some of the common features. Before I move on, I wanted to know if you guys know what the smallest animal is in the world. Do you guys know? Or what do you guys think? Maybe give me a guess if you don't know. You can type it or you can say it. You have to participate. Take a guess, maybe. Choose Devin, Audrey, Nancy, Bugbear. Let's see. Heard that name, maybe. Oh. Did you mean Big Bear? Have you guys heard about tardigrade? Tardigrade is water bear, known as water bear. No? So let me show you skin mites first. So skin mites are really small. They are on your skin and you don't even know. Sometimes they can give you infection, but they're really, really small in size. And then there is something called tardigrade. I think there's a cartoon too of this. This is the smallest animal in the world. This is known as water bear. So it's known as water bear. It dries out, and then when it gets some water, then it comes back to life. It's known as water bear. This is the smallest animal in the world. It's so small. It's even smaller than those single cell organisms we talked about, like diatoms and dinoflagellates. And this is multicellular. That's why it's considered an animal, even though it's smaller than diatoms. So let me see if I can show you the size. So here is a mite, dust mite. That's the size for a dust mite. This is a tardigrade. So it's really small in size. So this would be the smallest animal in the world. I'll be showing you a lot of different kinds today because we're not meeting in the lab, so I can't actually show you the actual specimens we have. So first animals arose from aquatic protista about 570 million years ago. If you think about Protista, we went over LJ when we talked about Protista. We went over protozoans. So protozoans, if they're single cell, they behave like animal, they're Protista. But when they're multicellular and they're swimming around eating other things, then they are considered animals. So the first animals arose from Protista. And so we had Protista, then it went to plants, algae became some land plants, and then Protista also diversified into animals. And then many animal phyla originated during Cambrian explosion. So during Cambrian explosion, the animals that existed then were wiped out. They went extinct, like trilobites. Or maybe animal with a different body plan, maybe with 5 legs. They don't exist now. So after the Cambrian explosion, it was a mass extinction. And also the ice age happened right before. So there was lots of habitat available. There was less competition. So whichever were able to survive, they diversified. So many animal phyla originated during Cambrian explosion. And then it started to diversify because the ice age was right before Cambrian explosion as well. So animal features reflect shared ancestry. So ancestral Protista here, maybe it was single cell first. And then, so it was a Protista, ancestor single cell Protista. And then it started to turn into clonal cells, means the cells started working together. Then it turned into multicellular. And then it became animals. So multicellularity is one common feature that's shared by all these different phyla of animals. We have 9 major phyla. So I told you the mnemonic, kings play chess on fine green squares. So we have kingdom, Animalia. Under that, we have 9 major phyla that you guys have to know. This is not the only phyla that exists under kingdom Animalia. There's a lot more. There's even some it's so specific, there's only one animal in one phyla. So there's a lot more. If you're interested in learning more, you can take a zoology class, they'll go over that. But for this class, we're only going to cover the 9 that are listed here. So let's talk about some of the characteristics here. So first one, after multicellularity is shared by all animals. And then we have something that says no tissues or true tissues. No tissues mean it doesn't have nervous tissue, it doesn't have muscle tissue, no neurons. It's just a bunch of loose connective tissue working together, a bunch of cells working together, and that's sponges. So we have only one phyla under that, and that's sponges. So phyla is plural. Phylum, so we have only one phylum here, is singular, sponges. If it has true tissue, that means it has neurons, it has muscle tissue or some type of tissue, and everything else falls under true tissue. So all the other ones have true tissue. And then after that, it starts to split one more time. And here, the feature we have is bilateral symmetry plus 3 germ layers, and then radial symmetry and 2 germ layers. So we'll talk about that next. So first branching point in animal taxonomy distinguishes a clade with true tissue from a clade with no true tissues. That's the first one. So sponges go under no tissues, and then everything else is true tissues. So in sponges, cells do not interact. And they, to provide specific functions, they kind of just work together, and they don't have true tissues. I'll show you a video how they work together too. And then the second branching point is the symmetry. Symmetry in, if it has bilateral, it's going to have 3 germ layers. I'll tell you what those are as well. And then radial symmetry is 2 germ layers. So symmetry, if it has radial symmetry, it's like a pizza. As long as you go through the center, you can get equal halves or mirror image. So here's the center. So here's the center. And if you cut it like this, you have a mirror image, same as whatever is on this side is also on this side. If you cut it like this, as long as you're going through center, it's going to give you the same right as the left. So that's radial symmetry. Any plane passing through the body from mouth to the opposite end creates a mirror image. This is a mouth, this is a hydra. And then there is bilateral symmetry. So to get a right and a left, that's a mirror image. You have to cut from head to toe. That's bilateral symmetry. So if you cut it like this in half, it's not going to give you a mirror image. You're not going to have, you know, you have 4 legs here. You're not going to have those here. You don't have the claws there. It doesn't give you a mirror image. So you have to cut it from head to toe to get an equal right and left. That's bilateral symmetry. So bilateral symmetry has only one plane. There's only one way you can cut it to get a mirror image. Does that make sense to you guys? Give me thumbs up or just tell me yes. Yes, okay. So on the exam, I can ask you, what are some of the benefits of having bilateral symmetry versus radial symmetry, or radial symmetry versus bilateral symmetry? Can you guys think of some benefits having bilateral symmetry or the radial symmetry? So radial symmetry is for the organisms that don't move around much. So it's a hydra, it's stuck to a rock basically. So something comes and touches it. It can feel in 360, can feel all around. So it's good for the animals that don't move around. So because if they can feel at 360 all around, they can get the information very quickly. But if you have bilateral symmetry, you're most likely moving around. It's very beneficial if you're moving around because you can get food, and being stuck to a rock is not good. It's not going to do much. You'll have to wait for the food to come to you. But if you have bilateral symmetry, it's going to go from head to toe, and you're going to notice all the sensory structures are in the head. That's where all the holes are: eyes, nose, ears. They're separated from the tail. So bilateral symmetry is going to have all the sensory organs in one place. And then that's going to make it very easy to move around because you have all the senses there. So if I ask you on the exam, what is the benefit of bilateral symmetry? You're going to tell me locomotion. Locomotion is moving. It allows animals to move around very easily. One thing, you have legs and arms, you can move around. You have a head and toe, and then you have all the senses in one place. It's going to help you move easily. So it's separated from the head and tail. That's why you can have the senses in one place. You can move around easily. You guys understand that? Does that make sense? So if I ask you benefits of bilateral symmetry, would you be able to answer that? Okay. Okay, so on the same branching point, I told you there's one more thing. It says 3 germ layers and then 2 germ layers. So that's germ layers in the embryonic development. So 2 embryonic germ layers with the 3. If it has a radial symmetry, which is only cnidarians have radial symmetry, it's going to have 2 germ layers. If it has a bilateral symmetry, which is everything else, it's going to have 3 germ layers. So let's talk about the germ layers. So in all phyla, except for sponges, there's embryonic ball of cells called a blastula. So if you know anything about embryo development, we've been talking about, you have an egg that gets fertilized and then it starts to divide, mitosis. It divides and divides and it turns into this ball called blastula. So it turns into this ball called blastula. And this ball is hollow. And because it's hollow, it's going to start folding onto itself. So it starts to fold onto itself. Once it does that, it's called gastrula. That's what you're going to call it, gastrula. So it's a hollow egg that starts to fold onto itself, forming a gastrula. In some animals, the gastrula only develops into 2 tissue layers. When it starts to fold onto itself, you call this inner layer endoderm, outer layer ectoderm. If it has radial symmetry, it's only going to have endoderm and ectoderm. If it's bilateral symmetry, it's going to have endoderm, ectoderm, and mesoderm. It's going to have 3 layers. The third one is mesoderm for bilateral symmetry. So again, radial symmetry 2 layers, endoderm and ectoderm. Bilateral symmetry 3 layers, endoderm, ectoderm, and mesoderm. So know these terms, know what the gastrula is, know what blastula is, and then endoderm and ectoderm and mesoderm, so 3 layers. Now, in the embryonic development, you can also see which body part is going to arise first, if it's going to be mouth or anus. So that determines if they are protostomes or deuterostomes. So that's what we're going to talk about next. So what we are doing, we're just talking about different features. Once we understand what these features are, then we will start talking about each phyla. Okay, if when the blastula starts to fold onto itself and turns into gastrula, and that opening, the first opening, if it becomes the mouth, it's a protostome. Proto means first. So if you're taking notes, you can say proto means first, stome means mouth or stomach. So the first opening becomes the mouth. It's a protostome. Now, if the first opening becomes anus, it's a deuterostome. Deutero means second. So so first opening becomes anus, and on the other side, the second opening becomes the mouth. It's a deuterostome. So those are 2 different types of developments. And you can't just tell by looking at an animal if it's a protostome or deuterostome. You have to look in the embryonic development. You have to watch the embryo grow and see which one becomes anus and which opening becomes mouth. So if first one becomes mouth, it's a protostome. If the second one becomes mouth, it's a deuterostome. Does that make sense? Okay. Okay, so next one we have, so here's a protostome. We have these under protostomes. In deuterostomes, we have echinodermata and chordates. That's us, chordates. Everything else, flatworms, mollusks, annelids, roundworms, and arthropods are protostomes. And then another thing we look at is digestive tract and segmentation. So digestive tract and segmentation, so let me show you an example first, actually. So body cavity means space. So if you get a cavity in your teeth, you have some space in there, and it's known as coelom. Coelom is body cavity, space, or a cave. So animals, they can have a bilateral symmetrical animal. So if it has bilateral symmetry, it can have a coelom. It can have no coelom, no cavity, or it can have a pseudocoelom. Do you guys know what pseudo means?
No.
Pseudo.
Fake.
Fake or false. So it's called a pseudocoelom because if you look at the coelom here, you have this cavity here, and it has this tissue layer, mesoderm on the endoderm, and then it has this tissue layer on the ectoderm as well. So it has tissue layer on both, mesoderm on the outer layer and also on the inner layer, and then a cavity in between. So this is a true coelom. It has a coelom. And pseudocoelom, it has mesoderm on the outer layer, but there's no mesoderm on the inner layer. So this is called pseudocoelom. Whereas for the no coelom, there's no space for the organs to move around, for the digestive organs to move around. This is planaria. This is planaria. And I'll show you more pictures of this. So coelom is a body cavity surrounded by all sides, surrounded on all sides by mesoderm. You have mesoderms on all sides. A pseudocoelom is surrounded on one side by mesoderm and the other side by endoderm. There's no mesoderm on the inner layer here. And then there is no coelom. So these animals, they lack a body cavity. There is no space for digestive organs. Or if they are there, then there's no space for them to move around. So let me show you the digestive system now. So some animals have incomplete digestive tract and some have complete digestive tract. I want to show you the complete first so the incomplete makes sense to you. So this is us. We eat, we put food in our mouth, goes through the esophagus, goes to your stomach, then your small intestine, and then your large intestine, and then out through the rectum, and then through the anus it leaves. So you have the entry point here, and then you have the exit here. Food is only moving in one direction. It's not going back or going up. So it's only moving in one direction. You have an entry point, then you have an exit point. You have your all other organs, liver, pancreas, and for digestive juices that mix the food, but it's still not moving in any other direction. It's moving in one direction. That means you have a complete digestive tract if food passes in one direction, mouth to anus. So if that's a complete digestive tract, this is incomplete. There's only one opening, and that opening is where this organism is going to eat food, put the food in, digest it, and then eject the waste out through the same opening. That's incomplete digestive tract. If the mouth both takes in food and ejects waste. I don't want to call it mouth because it's also ejecting waste. So I want to call it oral opening. So I'm going to say it says oral opening. It's a sac-like digestive system. It's a sac-like. Food comes in, gets digested, and the waste ejects out. So that's incomplete digestive tract. Does that make sense? Yes.
Yes, ma'am.
Okay, and next thing we look at is segmentation. If you look at this millipede, you can see same segments repeating. Here, centipede, same segments repeating. You guys know what number is milli? What number is milli? And what number is centi? Kawa, what number is milli? Milli is 1,000, and then centi is 100. Yes, so centi is 100 and milli is 1,000. Ped means feet, pedestrian or podiatrist. So ped means feet. These are known as myriapods. Myria means many, pods again feet. They have many feet, but then we call them millipede and centipede. Centi means 100, but it doesn't. If you sit down and count these, they don't really have 100 legs. It's just a guess. And milli means 1,000. It doesn't have 1,000 legs. It's just a random name they gave because it looked like there were a lot. So this is a millipede and centipede. They have segmentation going on. So the same segments are repeating here. This centipede, it has one pair of legs per segment. So if you look, it has one pair of legs per segment. So here. And then this milli has 2 pairs of legs per segment. These centipedes, they're carnivores. They eat other organisms. Millipedes, they're vegetarians. They decompose matter or eat decomposing matter. So these repeating segments, they can be modified into specialized body parts. Think about maybe insects. You can have wings on one segment. You can have a pair of legs on the next segment. So you can specialize these repeating body parts. It's going to give you more flexibility, and it has more potential for developing specialized body parts. So that would be the benefit of having segmented bodies. Gives you flexibility and you can specialize into different body parts. So different combinations of these qualities, so the ones we talked about so far, characterize each phylum. So now we're going to go into each phylum and see which type of features they have, these organisms have in that phylum. So that's what we're gonna do. Now, this chapter is a lot of memorization. You can make flashcards or you can create a spreadsheet like this. So I decided to do an Excel spreadsheet. You can list the phylum, common name, symmetry, digestive system, the type of digestive system it has, some unique characteristics, and then some examples. If you want to do a spreadsheet, you can do the spreadsheet, but I recommend that you write it by hand because you have muscle memory. You will memorize it a little bit better if you handwrite it, but you can do either or, whatever works better for you. If you need more motivation to study this different animal phyla, you can pretend to be a tour guide at Monterey Bay Aquarium. They have all these different unique characteristics about different phyla memorized. And when the guests come through or a school group comes through, they start telling them and they ask, oh, what do you think? The sponge is a plant or an animal? So they have these different phyla memorized. So you can try to do that if you ever think about being a tour guide or something. So that's what we're going to do now. We're going to start going over this different phyla. So first one we have is sponges. So let's look at sponges.